Mm. Hello. Hello, Howard.
Hello, Victoria.
Science
How are you?
Society. Great. Glad to see you here.
Yep. How's the audio?
The audio is fabulous.
Okay, very good.
I'm just calling Katarina in here and some other people. You are the early bird. How's your day going?
Pretty good. Where are you in the world? Are you East Coast or someplace else?
I'm right now in Oregon.
Oh, very good. Okay,
Mm-hmm.
so my day is on the closer to the end, I should say. So we're wrapping up stuff.
So you're on the East Coast, perhaps?
Yep. Mm-hmm.
Any exciting weather? I hope you're not bracing for a hurricane or anything.
No, it's gorgeous outside right now. It's gotten cooler. We've hit autumn officially, and we had the warmer weather maybe like 2 weeks ago, and then it was like a switch, and then bam, now it's cooler.
Yeah, that happened here. We just got our first rain, first of many, but,
Mm-hmm.
but it's okay because that's why it's so pretty, and keeps the slugs happy.
Mm-hmm. Are you whereabouts in Oregon are you?
Hi. Hi,
Katarina.
Howard. Hi,
Hello,
Victoria.
Katarina.
Hi. So Howard,
Sorry that I was kind of late. I wanted to be 5 minutes earlier, but I'm a minute late.
we
So
didn't even mention it. We
thank
knew
you.
you were on your way. But Howard, do you mind clicking on our biopics here, and
Sure.
then when you see make moderator, do you mind doing that,
Oh,
please?
sure. Yeah. No
Then
problem.
we have the magic powers so that you don't have to worry about running the room. Yay! Well done. Thank you.
Okay. Very
Thank
good.
you, Howard.
No
How
problem.
are you?
Good. How are you?
I'm good, good. Thank you. Thanks for opening the room and everything. I will put the link on top of the room to the paper, unless you want to start with maybe your lab website. I also have it ready.
Sure. It's up to you.
Okay, then let's
The lab
during the
website has a nice picture.
perfect. Can everyone see it? Yeah. Oh. Usually there's a picture on the left side, but somehow it's not.
I wish we could see that.
Right?
Yeah, it would be so nice.
Let me also put it in the chat so people have later access to it. And the paper.
What is the photo that we're not seeing?
I guess it depends on if there is a logo for the website, like a Famicom. Maybe it shows up. But if you click on it, let's see, we got plenty of photos.
So yeah, welcome everyone. Hi Einar, hi Soil, hi Manas. Feel free to raise your hand, come up to the stage, participate, and then if you think this is interesting, please feel free to also share the room. And yeah, we'll start on top of the hour. So a minute left for me to share it on Twitter. Sorry.
Right, it's that housekeeping that we're not able to do until we're actually here in the room.
Okay. Yeah, I think we can slowly start, and people will continue coming in. But since we start with the introduction for a little bit, I think we can go ahead. The recording, having the recording in mind, because we have friends all over the world, and there's never a time that works for everyone. So people will have the recording. So, welcome everyone to Science Society, and of course, a special welcome to you, Howard. Thank you so much for taking the time to come here and speak about your really interesting research. And everyone, if you're interested to learn some more, we on top of the room the Salis website. There you can find more resources about different publications, and also lab members and what the lab is about. So feel free to check it out. It's a really cool, pretty website that sadly the picture doesn't show. And before we start, let me give you some information about our guest speaker, Dr. Howard Salis. He's an associate professor of biological and chemical engineering and synthetic biology at and his areas of expertise, or the areas he is working on in his lab, is synthetic biology, metabolic engineering, biophysics, molecular biology, and biochemistry, modeling and optimization. And Dr. Salis, he did his Bachelor in Science in Chemical Engineering at Rutgers University, and then his PhD at the University of Minnesota in Chemical Engineering. And he then did his postdoctoral research at the University of California in San Francisco in the Voigt Lab. And he's also the founder of De Novo DNA, who is a spin-off company from the Salis lab at Penn State University. That's really interesting. For sure, we would like to hear also about your company if you have time. And now he is, as I said, an associate professor at Penn State University. And he won different honors and awards, including the NSF Career Award and the DARPA Young Faculty Award. And so, yeah, we are very honored to having you here. And usually, Victoria asks like a couple of interview questions so people get to know you a little bit better. So,
Sure.
yeah, thank you, Howard.
Thank you very much for the invitation to speak with you all today.
Thank you, Katarina. Howard Science Society is so happy to welcome you here. And that was really exciting hearing about all the cool bio things that you've been up to in your life. And it would also be great to hear a little bit about your background as we prepare to hear about your talk, because Science Society is all about people exploring knowledge together. So we appreciate knowing about the person who's about to present. So my question is, if you can tell us, if you think back in your lifetime, when, if there was such a time that you noticed a spark that made you feel like you had an affinity for science. And that could be any time in your life, little bitty time or more recent.
Sure. I guess I would say that from a very early age, I always kind of knew that I wanted to go into R&D, research and development. In fact, my mom would always say, "Oh, you were talking about this when I was in elementary school," let's say. And my father is an engineer, my mother is a nurse. So, both of them were very much encouraging of going into the sciences and engineering. And so from a very, young age, I was exposed to, computers. and I got to program computers, and I learned several languages. Was also very immensely interested in biology and microbiology during high school, and so when I entered college, I basically wanted to combine those two passions, so computers and biology.
Right. Thank you. It's really interesting to notice that you can take what your previous interests were and then combine them maybe into a new field of study or research. And so perhaps can you bring us from the point that when you entered college and you were interested in microbiology and other bio fields, can you bring us up to the point where you're now at your current research, maybe some of the larger events that happened, something to bring or
Sure.
along your journey? Thank you.
So maybe my second week of being an undergraduate student at Rutgers University, I walked into a tissue engineering lab. So this is, circa 1994, when tissue engineering as a field was just beginning. And I walked into a faculty lab, and I basically, as a freshman, I said, I'll do anything. I will take out the garbage if you want me to, but I want to, work in your lab. And so he looked at me and he said, all right, I'm going to put you to work. And so I was trained quite a bit with tissue culture. So for those in the clubhouse room who know, um, you have to be very detail oriented, very careful about your movements whenever you do tissue culture. And we were working with epithelial skin cells, and so they are adherent, and so there's lots of extra steps to culturing and subculturing and basically babysitting those cells. And so as a young student, I got into the rhythm of doing research, um, and really scheduling into into my curriculum that I would be in the lab at this time at this day, and I would be assisting a graduate student on their project. And then of course, as I got older, I wanted to work on my own projects, and I had in my mind the types of projects that I wanted to work on were really more towards um, this idea that we as engineers we design things on a computer and then we build them in real life. So we design cars on a computer, we design bridges and trains and planes on a computer, chemical factories on a computer, but we don't, particularly at the time, we never designed organisms on a computer. That was still science fiction. And I wanted to make that a reality. I wanted to sit down at a computer and design an organism from the bottom up, and then go into the lab and build that organism and write out its DNA sequence, physically construct that DNA sequence, put the DNA into the organism. I wanted to make that happen, and I wanted my model predictions to be so accurate that we could create a whole new engineering discipline around organism engineering. So from a pretty young age, I was like, why isn't this a thing yet? And why can't we do this? And so I wanted to, so my roundabout way of doing that was to get more involved on the computational side and become more of an expert in computational modeling. I worked in a fluid mechanics lab that had nothing to do with biology, but they worked on very complicated fluid flow systems. And I did experiments actually testing those predictions. And I worked I took all sorts of courses in nonlinear dynamics and chaos, and got very interested in all the different types of modeling that one can do. And then in graduate school, I got very involved in what are called stochastic models. So this is where probability theory comes into play, which is important for biology because when cells are very small, and the number of molecules inside of a cell is also very small, compared to a mole of molecules. And so when you actually go to write down equations for describing what is happening inside of a cell, well, you're not dealing with a deterministic system anymore. You're now dealing with probability theory. The cell is effectively rolling the dice on how often, let's say, a transcription factor binds to a DNA site and how often transcription takes place and so on. And because it's constantly rolling of the dice, you can now have multiple things that are possible. So multiple different trajectories of your system are possible. And that actually has a big impact on how cells work. And so from a fundamental basis, if we want to make predictions about biology, they can't just be the same type of predictions that one would use, let's say, for a mechanical system with very large moving parts. The types of equations that we need to use to make predictions about biology have to be at the molecular level, taking into account probability theory, because everything is so small. And so I got really into that and during graduate school. And it got very detailed, very in-depth in terms of how do you write out the equations, how do you solve these equations on supercomputers to make predictions about biology and genetic circuits and genetic circuit regulation. And then, let's say I had my own little existential crisis near the end of my graduate school, because even though we were making lots of predictions about what was happening, we didn't yet have, let's say, the tools or the throughput to test all those predictions. And we couldn't still make predictions in the same way that we could with other types of systems. So in terms of like accuracy. And so during my postdoc, I basically wanted to solve that problem. I wanted to try to like connect these two worlds. Like, before, circa 2008, 2007, there was the systems biology world. Everybody was very much interested in writing out equations and solving them, making sense of systems. Then there was the genetic engineering world, where people were cutting and pasting DNA and trying to get a small genetic circuit or pathway to work. I wanted to like combine these two fields together and be able to make predictions, but not just make any prediction. I wanted to be able to predict the specific DNA sequences that we should be introducing into an organism to make that organism do something that we wanted. And that's a conceptual leap, because before, again, in the old days, we were just dealing with equations and numbers and making predictions about what the system would do. But now I wanted to go from DNA sequences to equations and numbers and predictions. And I also wanted to go from backwards. I wanted to go back from functions of interest to, through equations and numbers, but ultimately designing DNA sequences. And so conceptually, going from sequences to numbers and numbers to sequences back, that's pretty difficult to do, because a DNA sequence is a string of letters, but that string of letters controls gene expression through a whole compendium of different molecular interactions. And so in order to go from sequences to numbers and then from numbers back to sequences, we need to really understand at a very quantitative and predictive fashion, we need to understand how the sequences control all the things that can affect function. So the sequence function relationship. And the way that I went about doing that, understanding that relationship, is to understand the biophysics taking place. And so during my postdoc, we developed the very first algorithm, model, that could take in any sequence and make a strong prediction about what the function of that sequence is on gene expression. So this was the ribosome binding site calculator. You can input in any messenger RNA sequence, and it would predict translation initiation rates from that sequence. And you can go backwards. You could say, all right, I want a sequence that gives you a translation initiation rate of, let's say, 10,000 on a proportional scale. And you would enter in a number, 10,000, into the algorithm, and it would then go through a series of calculations and design for you a messenger RNA sequence that would give you that number. So again, sequence to numbers and then numbers back to sequence. And with that capability, it really unlocks, synthetic biology as an engineering discipline. So you're not just cutting and pasting DNA, you're not just copying, you're not just moving parts around, you are making predictions and you're carrying out rational design. Do we have any questions so far?
Well, Howard,
Yeah.
that was an
Yes.
amazing. That was like like listening to a sci-fi story. You're a really great storyteller, by the way. I'm
Oh,
sure everyone is thinking about this too, because you really carried us along through this whole process, and then and then you are developing a new discipline, and I want to hear the rest of what you have to say. So yeah, please. At this point, I just want to hand you the mic forever and ever, and and then you have your PDF there that you can lead us through, and or I guess your website. And then if you'd like to have Q&A at the end of your discussion, or have Q&A driving your discussion, that's completely up to you. And then I see you've already visited the room chat. And so people may put questions or comments in there, and you're also welcome to put or ask us to put a link in there that, if something comes to mind. So, but we're here to moderate the room for you, and thank you so much. And so at this point, the mic is yours. Thank you.
Okay, thank you. Well, it's always, I'm a faculty, I teach courses. I always like to get feedback from the audience about what is it that they care about, because of course I can go on for a very long time about our work and particularly this most recent work. But I want to hear from the audience, if they're so willing to speak up, that what do they care about? What areas of what topical areas of science are they interested in, and how there's quite a lot of intersections. So for example, our work helps a lot of people by enabling people to design and control gene expression for all sorts of different biotech applications. And we collaborate quite a bit through, partly through our company to help people and enable them to pursue those biotech applications. But I want to hear from the audience about what they're interested in before I can, I go off on my own tangent.
Yeah, thank you so much, Howard and Dennis. And so here you joined the stage. So if you want to comment or ask something, please go ahead.
Thank you, Katerina. Thank you, Howard. I'd be very interested to understand the implications of your work for the situation of COVID.
Sure. So here's one example. When people are developing the COVID vaccine, it's a mRNA vaccine. It's produced inside what is called an in vitro transcription assay. Our model predictions can help people make sure that when they encode the mRNA vaccine, not just the vaccine itself, but the genetic parts that are being used to produce the vaccine, that those genetic parts have, here a high transcription rate, producing a lot of the mRNA vaccine, and also at the same time, the system is not producing other types of RNA that is undesired. Okay, so in that regard, it's like, if you think about computational design and for organism engineering, our models and algorithms allow people to carry out their desired function or desired activity while not having to worry too much about the nitty-gritty details. So you can get a little bit, we've done, we've taken that effort, we've done all the experiments to develop these models and validate them so that other people, when they want to go work on a particular application, they don't need to, take into account the 30 something whatever different interactions that are important. our model is calculating the strengths of those interactions and reporting them to the user. And so they can just see what is happening, and they know ahead of time what is going to happen.
Thank you. Who would like to go next?
May I ask? Is it my turn?
Sure.
Yeah, thank you. So, in my understanding, problems such as protein folding, they're hard computational problems. So, is it that do you have like a solution to have a kind of solve them, or is it an approximation, or maybe you don't even, or do you actually kind of plan to actually create them in reality? Is it just for later on moving on to uh, implement them using the DNAs or not? And then my third question, which is actually my main question, is how much these genes and interactions and what a design mRNA does is computable in principle. So how much are we in our with us humans with our computational um, tools and understandings of biochemistry, etc., how can we, in principle, predict what a genome does if we have a sequence? Thank you.
Very good questions. So I'll answer the first one first. So my best analogy, because you brought up protein folding. So think about a car and all of the different parts in a car. So, you can be an expert on just the engine. You can just design engines, and you can just tune that engine to get the best gas mileage, let's say, or a hybrid. But that engine is just one component in the whole automobile. And so when you go to actually design the whole car on a computer, you're really taking into account all the different systems and subsystems, how they all interact, how they all connect to one another. And so the way to view what my lab does is that a lot of what an organism does is not just one part. It's actually like 100 different parts all working together, and that's just for one engineered function that we're adding into the organism. If you were to look at all the functions that the organism does, we're talking about, thousands of parts all working together. So if we start off with an organism, we say, all right, we want to augment that organism with a new capability. That could mean adding in 20 different proteins. And those 20 proteins could originate from many other different organisms. So these are like, recombinant or heterologous proteins that we are introducing into this organism. And then on top of that, we're adding in a whole bunch of new genetic parts to control the expression or production rate of these proteins inside the organism. And that control might be, it could be always on. So these proteins might always be produced under a particular condition. Or we might want to turn the production of these proteins on or off, or tune the production rate like a dial according to different conditions. And that can become important depending upon the application. Like, for example, let's say you want to feed the organism some low-cost feedstock, let's say like carbon dioxide, and you then correspondingly want to produce a higher value product that is needed by human civilization. Let's say how about a plastic monomer. And so you are engineering that organism to convert the CO2 into the plastic monomer. But in order to do that, we're introducing in, those 20 different enzymes, which is a type of protein, and we're introducing in all those genetic parts for controlling the production rates of those enzymes to make that conversion possible. So that is the type of organism engineering that we specialize in, because we don't focus necessarily on just the one protein. We look at the whole system, and we engineer the whole system to maximize the function of the organism. So in the case of the CO2 to plastic monomer, we want to produce the most amount of that plastic monomer as possible, not just a little bit. And then to your second question, how general are our predictions? That is, how good are we? What is the state of the art? So the state of the art in our field is that let's say for bacteria, which are simpler, we can input in the sequence of that bacteria, the genome, and we can predict the transcription initiation rates, the translation rates, and the messenger RNA decay rates. And we can predict some other characteristics of the sequence, like its copy number, depending upon where it is in the genome. And we can combine all those predictions together to get a pretty good prediction for how much protein is being produced, like straight just from the sequence. And then on top of that, we can make even better predictions about what happens if we make a modification to the sequence. So we know ahead of time that if we change, let's say, this G to a C or this A to a T at the DNA level, we know ahead of time that the transcription rate might increase by a factor of 2, or the translation rate might decrease by a factor of 5. And it's actually easier to make those types of differential predictions because you're starting with some sequence, and then all we need to do is predict the change in interactions when we make a modification. And so the state of the art is we're really good at predicting how a change in the sequence will affect the organism's function for bacteria. And that ability really drives the development of all these different biotech applications, because we can't always, when you're engineering these organisms, it's kind of like throwing darts at a board. And if we're able to throw a dart and hit the bullseye on the very first try, I mean, that would be awesome, but we're not there yet. But right now, the state of the art is that we can throw 20 darts for a simple system, or 100 darts for a more complicated system. And we can, of course, throw those darts at the bullseye and try to hit the bullseye, and we're going to get very close. And through experimentation, we'll figure out how close we got to the bullseye. But with the model predictions and our design algorithms, we know ahead of time that if we throw a small number of darts, we're going to hit very close to the bullseye and get the job done. So that is the state of the art right now. Next question.
May I ask the next question?
Sure.
Okay. So I understand that you have this whole system. It's like you have so many promoters you're looking at, and you give a whole output. For example, if I'm only focusing on one particular, let's say RNA polymerase, and I want to make changes. So your system would give some evaluation, says, let's say I'm going to do that, I don't know, amino acids like 350. And would that be a better changes versus another location amino acids, let's say promoters, or maybe let's say three prime UTRs. So if I ask you, say, please look at this, I'm working on this molecule, how I'm going to do, I want the output of a particular protein to be 2 times or 5 times more than normal, so that your system can help. Am I understanding, is it in this way, or am I going not quite the way
you're on the
yeah.
Yep, you are on the right track. Let me, because you brought up the paper, why don't I just give a 5-minute
Yes,
overview of the
please,
paper
please.
itself.
Yes, I was looking. Yeah, I
Uh,
was trying. Okay.
no, sometimes, you don't know. Some audiences are more interested in transcription versus others. So I didn't want to just jump into the paper, but now it's a good time. Um, so this paper, which appeared in Nature Communications in September, our mission for this paper is to be able to enter any DNA sequence into our model and make a really good prediction about the transcription initiation rate for every possible start site in that DNA sequence. And to be clear, this is in bacteria, and we carried out this model development and validation for what is called the sigma 70 factor, which is the housekeeping sigma factor in E. coli. And this is important because the sigma 70 factor is the most predominant and most used sigma factor for transcription initiation. Okay, so the way that we developed this predictive model is we designed 14,206 promoter sequences. We constructed all of those promoter sequences, so physically, and then we characterized and measured the transcription initiation rates from all of those promoter sequences all in one experiment. And then we, of course, repeated that experiment, in triplicate to get, great data. And we also carried out what is called transcriptional start site mapping on all of these different 14,206 promoters to figure out exactly where transcription initiated on these DNA sequences. And so that's the data that we collected. And then we utilized all of that data to then parameterize a predictive biophysical model of transcriptional initiation that then allows us to make these predictions moving forward. And so the sequence space that we're talking about here is, quite large. A core promoter sequence in bacteria is on the order of, some people would say 60 base pairs. We would say it's actually quite, longer, 80, 85 base pairs. And so if you look at the sequence space, it's 4 to the 80. Okay, so that's already an astronomical number of different DNA sequences. But that's what we are, that's the challenge. We want to make a prediction about what the transcription initiation rate will be given any possible 80 base pair DNA sequence. And then we actually show that you can take that model and scan it, use it to scan across a very long DNA sequence, like a whole genome, and make predictions, accurate predictions across the very long DNA sequence. Okay. And so in that first step, the way that we designed those 14,206 different promoters, it wasn't random. We first started off by reading a whole bunch of papers about what controls transcription initiation, from sigma 70 promoters. We then systematically designed sequences to perturb those interactions. And then we, combined all of those sequences together. It's not a combinatorial space that would be too large. It's a systematically designed set of sequences to perturb the individual interactions so that we can precisely measure the change in interaction strength with respect to sequence change. Okay. And then again, the way that we do DNA-seq and RNA-seq, which is a way of using next generation sequencing to measure mRNA levels in this assay. And in particular, we set up these assays to be fully in vitro. That is, there's only 4 ingredients in our assays. There's RNA polymerase and sigma 70. There's the library of 14,000 different promoter sequences. There's the NTPs, the fuel for transcription. And then there's the buffer in the water. And because there's only those 4 ingredients in our assay, we know that our sequences are not going to affect other interactions, like, for example, mRNA decay or transcription factor binding. And so when we carry out these measurements, we know definitively that the differences in sequence that we introduced will only have an effect on the RNA polymerase sigma 70's binding to these promoter sequences. And so that definitiveness means that our data is really good data that we can then use to create a really good model. And, you know, in the modeling world, some people just want to collect a whole bunch of data and they think that after the fact, they're going to create a really good model. It doesn't actually work like that. You need to collect really good data to generate and validate a really good model. And so we spent a long time making sure that the sequences that we designed are perturbing all of these interactions, and the assay itself was set up so that we were only measuring the thing that we wanted to measure, and other interactions were just not present and therefore could not confound our measurement. And so if you go through the paper, it talks a bit about this, but at the end of the day, the key benefit of our work is that you can go to one of our tables and you can look at the DNA motifs, and it shows you the interaction energies for each of the DNA motifs. Okay, which means that it's a fully human understandable model. You can look at this table and you can see what makes a promoter bind to RNA polymerase and sigma 70. You can look at this table and do a comparison across different motifs, and you can actually use the table without a computer, and you can design your own promoter. It's that understandable. Okay, it's like the opposite of all of these different like black box machine learning models, where you might train the model and test the model and all that, but you actually don't understand what's inside the model. Here, we do use machine learning, but we use it in a way where the output is completely understandable by humans. And you don't need to use machine learning afterwards to make the predictions. Okay? And I think that's an important distinction, because having been in the field for a long enough period of time and seen lots of different machine learning models that have been developed, a machine learning model might get a great prediction for a particular data set, but then as people use that machine learning model in a different context, a different system, it basically often, not all the time, but very often does not give a great prediction. That is, a black box machine learning model is often really just so specialized for a particular system. And you can't extrapolate. But here in our system, the way that we've set up the data analysis and the machine learning, the output of what we're generating is something that generalizes across any possible DNA sequence in bacteria for sigma 70, and allows you to make accurate predictions across the whole sequence space. And it's something that is human understandable. You know exactly what the calculation is and what you get out of it. Okay, and so at the end of the day, what does that mean? It means you can go to our website, you can copy paste in a DNA sequence, the model runs, it scans across, and it predicts a transcription initiation rate for every position in that DNA sequence. And it will tell you how strong your promoter is, but it also tell you, for example, how many alternative promoters or cryptic promoters there are in your system. And that's important because when we think of a DNA sequence as being a really strong promoter, oftentimes it's actually not one transcriptional start site. It's actually multiple transcriptional start sites that each bind separately and distinctly to different copies of RNA polymerase and give you a lot of messenger RNA. But it's not just one promoter. It could actually be multiple promoters combined together. And we're now understanding a lot more about how natural systems work and how we can engineer new systems with these model predictions, because the details that are being calculated and reported to you are now very fine and tell you quite a bit about what is happening at the molecular level. Okay, so I think that's a great stopping point for questions.
Yeah, thank you. Aina, did you want to go? Go ahead.
Yeah, please. Thank you very much, Katerina. So, if I get this right, the RNA will affect the DNA, and also then on go to the epigenetic part of the body. Is there any related research or science when it comes to what kind of what you put into your body or what you are being exposed of by the environment, which actually affects the epigenetic part of the time for when this will change the DNA? Yeah, thank you.
Okay, so good question. So let me give you an example of how our models and algorithms are being used to treat human disease. So and again, companies will use our models and algorithms for their own biotech applications. We have quite a few companies doing that. One of them, just a good example, is named Synlogic. And they engineer probiotic bacteria. And so these are, harmless bacteria that normally live in the human gut. One of them is called E. coli Nissl 1917. It was isolated in 1917. And they've engineered this E. coli Nissl 1917 to treat different types of metabolic diseases. And these diseases occur when a person doesn't naturally produce enough enzyme to break down a metabolite, and that metabolite then can actually accumulate in your blood to toxic levels. So in the past, if somebody had one of these metabolic diseases, and there's quite a few of these specific to the metabolite, but I'm not going to go into the detail there. But like, let's say there's one of them where you can't eat enough like threonine or lysine. So basically, if you were to eat a food that had too much of these amino acids, the amino acid would actually accumulate in your blood at a toxic level, and it would harm your liver. And so in the past, the treatment for this disease would be simply to eat foods that didn't have these amino acids, or at least not at a plentiful level, which is very restrictive. But now, Synlogic has engineered a couple of these different probiotic bacteria that express an enzyme that breaks down the metabolite inside of the human gut so that it doesn't accumulate to a toxic level. And one way that they use our algorithms is to produce a lot of the enzyme in the gut so that the organism is doing its job, correctly. And you might imagine that, so from a treatment point of view, what does a person do? They take a pill. Inside the pill, there, like any probiotic, supplement, there is an engineered bacteria in the pill. The engineered bacteria, goes down into your gut, and maybe only, 1% of the bacteria in your gut is this one strain of engineered bacteria, E. coli Nissl 1917. But that 1% of engineered bacteria is producing quite a bit of this missing enzyme. And the missing enzyme breaks down the metabolite so it doesn't accumulate and it doesn't harm your liver. Okay? And so that is one collection of biotech applications that I think are pretty readily understandable, and how, you know, synthetic biology is really helping people treat human disease.
Yeah, thank you for the question and answering that question. Yeah, I think this is really fascinating, and you know, very helpful, especially for rare diseases. With this model that you designed, so are there a lot of maybe universities or people coming with maybe how to solve or treat rare diseases, coming to you, to your company, and that they need help with that?
Yes, so we have over 10,000 registered users for our website. We and those users have designed over 850,000 different DNA sequences for a whole range of different applications. So the software is, you know, is used quite a bit, and it's, you know, many of those researchers are from academia. Others are from companies, and so we have quite a few companies, well over 100 right now. I'm actually looking up. We have a leaderboard. So if you wanted to know which university uses us the most, it is Harvard University. Has 90 researchers who have designed 99,782 DNA sequences with our software. The next one is Tianjin in China. The next one after that is Seoul University in South Korea. The next one after that is MIT, and so on and so forth. So it's very much international. You know, synthetic biology is a global field. Can you hear me?
Yes.
Okay, we're having a massive thunderstorm right now, and I think the, you know, reception is going to be affected. But okay, as long as you can hear me. All right. So you know, it's a global field, and you know, biotechnology is, you know, because it affects us in so many different ways. Of course, it's a global economic engine. So we collaborate with quite a few groups working to mitigate climate change, to break down plastics. We collaborate to, again, you know, engineer probiotic bacteria to express monoclonal antibodies and other recombinant proteins. We collaborate quite a bit with engineering metabolic pathways to produce specialty chemicals from low-cost feedstocks. So basically, sustainable manufacturing. In my own lab, we have academic lab. We have projects including engineering soil bacteria to detect the presence of explosives in the soil and report on their presence by producing a potent odorants to warn people about the presence of those explosives. And we also have other projects on, you know, engineering algae to grow faster. And that's part of a ambitious plan to sequester a gigaton of carbon per year, which we can't do alone. We need a whole big, large group of people to help us with, but we are using our algorithms and models to make that more of a reality. And then overall, you know, a lot of what we do is enabling. So when we develop a new model or a new algorithm that carries out, you know, improved predictions or predicts more of what's happening inside the organism, people will immediately start using that new model and new algorithm in their own application. And so like we'll develop something in the lab and test it and put it up on our website. And then, we'll see people immediately start using it. Like our recent paper that I just talked about has already been used to design or predict 22,000 different sequences. And again, the paper's only been published for a month now, although it was a preprint earlier. And so the benefit of what we do is that it immediately starts being used because we make it very user friendly. And then, as it happens, 2 to 3 years later, we find out what people are using it for. And so there's those thousands of applications, producing all sorts of different small molecules, plastic precursors, therapeutic candidates, vitamins, nutrient supplements. People are using our algorithms to produce all sorts of beneficial compounds.
Well, that is very impressive, and you really didn't just write in the grant that you will create a new research field to get these grants. You actually did, which which is a very big difference. I know we always write that to get those special grants, but if we then actually manage to do it, it's a whole different story. So it's so impressive and so beneficial for humanity what you have been doing. I'm recently really interested in, due to company that I work with and consult for immune responses and stem cell 3D bioprinting. Do you think your model could also predict more things like immune responses, or if you maybe change a gene that triggers some immune response, not just what effect it would have to change the genome that way, but from a stem cell, let's say from a donor, but also maybe what the immune response or the reaction of the whole organism would be? Do you think you can use the principle of the model to scale it to a whole organism or complex system? Like, I know it's already a complex system what you're doing, but you know what I mean, like a bigger organism or context type. Let's say you change the like, I'm giving just a few examples to explain it better. You change the microbiome. Would you maybe be able with some data to predict what it would do to the person, or if you would try to rescue soil after major forest fires, if you would implant microorganisms in the soil again, what the outcome would be? Do you think that is something your model could also predict?
Let me take that first question first. Okay, so you can hear me, right?
Yes, thanks.
Okay. So there are techniques for doing immune profiling, and we actually have my spin-off company has developed a new gene synthesis technique, which we call massively parallel protein libraries, where if you want to express 10,000 different proteins of interest, defined sequences, so defined amino acid sequences, we can now construct for you a library of those 10,000 different protein sequences, and they're all uniquely and individually barcoded with a unique DNA sequence. And then you can do effectively an antigen capture. So if you're expressing monoclonal antibodies, you do an antigen capture. If you are trying to figure out which antigen binds to a particular monoclonal or antibody, you can actually express 10,000 plus different antigens and then do the antibody capture. So what you can use that technology for is to actually figure out exactly what you said, the interactions between our immune system and a particular set of antigens, and figure out how mutations affect those interactions. So it's not a model, because I will say that we don't yet have enough data to make all of those types of predictions that we would need to do for de novo immune profiling. But what we've been able to do with my spin-off company, De Novo DNA, is to substantially, like 100x, reduce the cost of protein library construction, so that you can do those 10,000 plus different experiments all at the same time and collect all of the data at the same time for again 100x less fewer dollars compared to the existing state of the art.
Well,
So that's something that, yeah, go ahead.
yeah, that's amazing, because what the crucial step is to, let's say we can 3D print like complex systems, which is a challenge by itself, but sometimes we can, right? So some of them we can. The thing is, it's way too expensive to make an organ for every single person, a specific one, grow the cells from that person, and so on. So what would make it scalable and affordable for the large population is to have off-the-shelf organs. And for that, we would need to know, for which cell type, what we have to take away or add, what type of proteins, because sometimes also just adding signals, I'm fine, you can leave me alone, I belong here, type of thing, that would be hopefully enough to not have major immune response issues. So that would make the whole industry scalable, and probably you're the closest one to be able to predict that, to cost and able to design that product, that off the shelf product. So that's really exciting.
Yeah, so we're providing this as a service through our company, where again, you pick your favorite 10,000 proteins. The restriction is 300 amino acids or fewer right now, and it's constructed in a tube and sent right out to you. So and the pricing it varies from $5 per variant at the 10,000 scale. And if you ask for fewer proteins, the price per variant does go up because there is economy of scale. But okay, so other questions? Kirko.
Hey Kyoko, do you want to ask a question, or or comment maybe?
Yeah, I do actually have a question. Um, I was curious, is it possible, or do you think like this would be too far-fetched that this technology could be used to predict short DNA fragments' ability to form like secondary structures? You know, like RNA type deal, or even just like what I'm saying, like hairpins and stuff like that.
Absolutely. So we know quite a bit. Collectively, the whole field knows quite a bit about nucleic acid biophysics. So if you have a single strand of DNA, we can predict how likely it is to form all different types of structures, both secondary structures and even some tertiary structures, like including hairpins. We can do that for DNA. We can do that for RNA. We can do it for DNA RNA complexes. We can do that for multiple interacting DNA or RNA strands. And there's all these really cool nucleic acid genetic circuits where you can synthesize different types of DNA or RNA strands and then you can add in a new strand and trigger a chain reaction that changes the structures. And so there's all these like types of cool input-output circuits that one can build using purely short strands of DNA or RNA as the substrates and regulators. So the field has gotten pretty sophisticated in that regard, in being able to calculate and make these types of predictions. But of course, that's just DNA or RNA interacting. A lot of what we do is we take those same type of calculations and we then apply them to inside the cell, and what happens when DNA or RNA form structures inside of a cell, and how does that affect gene expression levels, for example, at the transcription rate step or the translation rate step. And I'm getting, yeah, oh, yep, thanks. And I'm getting some questions in the chat. Is it possible to make a library of mouse transcription vectors and AAV plasmids, and a library that enables cloning of different tags? Okay, yeah. So with this new technique that my spin-off company has developed, you pick your favorite 10,000 different proteins. Right now, fewer, 300 amino acids or fewer. Define sequences, so it could be any amino acid sequence, and we will, construct them and in vitro and create a plasmid library, and we then send that plasmid library to you. And it could be whatever vector that you'd like, within some size restriction. It could be AAV. And the spin-off company is called De Novo DNA. I'll just put it in the chat.
Hi, Dr. Cha. I know you just arrived, but do you have maybe a comment or question you would like to ask?
Hi, Katerina, and yes, I just missed this one. So yeah, I'm just take a look. If we have time, I'll be back to that. Otherwise, thank you so much for sharing your research with us.
You're very welcome.
Yeah, if we have some time in the meantime, um, if you did you have also included in the mechanisms these I-motif foldings? Like, can you predict how likely it is that there are this G4 or I-motifs or other type of foldings in the DNA under different like environmental factors?
Yes, so we have a pretty good understanding for what causes an I motif or a G quadruplex or other types of tertiary structures. And in our other work, not this this paper, but in our other work, we have actually measured how those different motifs affect mRNA decay rates. So we did find that G quadruplexes have a protective effect on messenger RNA and prevent them from being degraded inside of cells, whereas I motifs seemingly had no effect whatsoever on mRNA decay. They still decayed. So that was interesting. So, we, collectively the field has a pretty good understanding for how a short motif folds into a particular structure. Then, with our work, we try to answer the follow-up question of how does that motif affect gene expression levels? Do you want that motif present in your sequences, or do you not want it because it has a negative effect? Or, how can we use that motif to tune expression, up or down according to our desired target? And with our other work, if you look on Google Scholar, you can read some recent papers where we've really figured out the rules for controlling mRNA decay rates as well. And some of the rules are quite simple. Like, for example, if you have a large unstructured region of RNA, then it becomes a target for RNase E, and that RNA will then be chopped up at a faster rate. And so if you want a higher mRNA decay rate, then of course you can design your sequences to have large unstructured RNA regions. But of course, if you want to have a really stable messenger RNA, then you design that sequence to actually fold into a structure to prevent RNase E from binding. And so a lot of our design rules work together so that when we design the system, we're making predictions for transcription rate, translation rate, mRNA decay rate. And a lot of these predictions are coupled together so that, when we make those calculations, we might make a sequence change in one location that actually influences all three different gene expression, processes at the same time. So it's important to be able to separately predict the effects of a sequence change on all these different coupled processes to make a really good prediction.
Yeah, that's really interesting. And would you be able to basically model what it would do to a neuron? Because I had this theory that I never got funded for, that basically eye motifs and so on play a role in acute plasticity and maybe even long-term plasticity in the neuron, like for memory formation, basically for short-term maybe. Would you be able to predict what the different cell types, how it would change the shape or membrane formation and so on with your model?
Well, again, so think about it this way. Our models are able to predict sequence function relationships, how changing a DNA sequence affects gene expression levels at the different steps. And those predictions are then used to engineer a large genetic system to carry out a desired functionality. And so that capability has been used for engineering microbes for all sorts of applications, whether it is to, for sustainable biomanufacturing, whether it is to treat human disease by engineering probiotic bacteria, or producing biomaterials to replace, plastics and materials that came from petrochemicals. So there's a whole economic sector that we are seeking to advance by developing an engineering discipline for organism engineering. So it's being able to engineer these organisms in a very rational, predictive, reliable way means that you can then creatively accomplish quite a lot without having to worry about, this interaction or that interaction or the coupling between them. So we are, putting on our engineer hats. We want to get to a point where we can go from, stating the problem to designing a solution really quickly on a computer, and we know that if we build it, it'll just work.
Yeah, thank you. That's that's really good for designing drugs and so on. Also, maybe for different type of disorders. So yeah, it's really wonderful. No, Dr. Shah, did you have a question in between?
Yes, so my question is about, because you just mentioned about the benefit of using that, and I was just wondering, for example, for the cancer patient, we are using the liquid biopsy, and we have a measurement of the mRNA out of the exosome. And because you just talk about the, different level with the bacteria, do you think that we can just use it for the, tumor detection or those kind of cancer development research or not?
So in the area of cancer, there are a lot of natural bacteria that produce chemotherapeutics. In fact, a lot of the chemotherapeutics that we're using right now If you're familiar with like vincristine or vinblastin, they all come from microbes. And so there's a lot of interest in being able to produce and synthesize these chemotherapeutics, not using synthetic chemistry, which makes them really expensive, but using engineered biochemistry to produce them in a more sustainable way and low cost way. Um, Here's one example. Taxol is a chemotherapeutic. Um, the yield for making Taxol is via synthetic chemistry is super small. It's like 0.0001%. That is, you start off with a kilogram of initial product, initial substrate, and you end up with a couple of milligrams of Taxol by the end of it. That's why Taxol is so expensive. But alternatively, you can engineer an organism to produce the compounds that go into Taxol synthesis, getting very close to the end of the pathway. And therefore, you can substantially increase the yield of production for Taxol so that you can drop the price of Taxol by several hundredfold. And that's important because it's not just about um, I mean, it's not just about developing these new drugs. It's also about making them much more affordable for people. As we know that healthcare costs are growing and becoming predominant, and so any way that we can take a drug that is already on the market and substantially reduce the cost of that drug is a big win for the field. And in particular, with synthetic biology and engineered biochemistry, if you start off with a pathway inside of a microbe that makes a drug of interest, it's actually really easy to introduce new enzymes into that organism to then make modified versions of that drug, and that those modifications can actually make even better drugs. So there's a lot of efforts in the field to start off with a drug of interest that is already on the market, but then with a couple of extra modifications and introductions of new enzymes into that organism, now you can create a completely new drug. It might not be a chemotherapeutic anymore. It could be it could be an anti-aging drug, because a lot of these functional groups are shared across different disease types. And so we're learning a lot more about how to sculpt and design drugs and work backwards to what enzymes need to be produced inside the organism to make them.
Yeah, for example, E. coli, that they are just using for example, expressing the cytolysine that they are using for tumor detection, or in some of the researchers, they are just injected to the tumor. And that was very beneficial. So I think that based upon your research, it would be much easier for, track the activity inside the tumor, because microenvironment of the tumor is really challengeable.
Yeah, there's a lot of really interesting work on using bacteria as like homing missiles for tumors because they naturally accumulate inside of tumors. I was involved in some really early work using salmonella to kill tumors, which has challenges because salmonella, of course, is a pathogen, but it's definitely harmless compared to cancer. Um, and more recently, there's been a lot of interesting work not with cancer, but with using engineered phage to kill pathogens. Phage are viruses that kill bacteria, and so nowadays, people are greatly worried about antibiotic resistance and the onset of bacteria with multiple antibiotic resistances. And there's actually quite a few pathogens that have been identified that are resistant to all of the common antibiotics that we normally would use. And so people are now using engineered phage and using our algorithms to design these phage to enter these pathogens and express proteins like lysins or barnase, which destroys DNA, to completely eradicate these pathogens. And so it's a new type of antimicrobial agent that can supplement antibiotics and prevent antibiotic resistance from happening.
What about yeast and fungi? Did you add any research around those as well?
Yep. So my lab does work in those areas. We have a recent paper where we designed and characterized 25,000 different, 1,700 different non-repetitive yeast promoters that vary transcription rate by 25,000 fold. So we were able to design these sequences to vary transcription factor binding and vary transcription rates by quite a lot. And through the design process, we made sure that one could actually utilize all these different promoters at the same time without introducing repetitive DNA into yeast. Because if you're not aware, yeast homologous recombination is super efficient. And so anytime anyone introduces a repetitive region inside of the yeast genome, well, that repetitive region will trigger homologous recombination, which will then delete the system of interest. And so we basically developed a solution to that problem by creating a new algorithm that designs non-repetitive genetic parts, including these non-repetitive yeast promoters, so that when you go to engineer the yeast, you're not ever introducing repetitive DNA.
Very interesting. And what is the treatop of them? For example, is it Saccharomyces cerevisiae or what?
It was
species?
Saccharomyces.
Yeah, I see. Thank you so much. Thank you for sharing your research with us. That was wonderful.
Thank you very much.
Yeah, I agree. Thank you so much for coming. I don't know if anyone has any last questions or comments they would like to add. I think you did such a wonderful job. You moderated everything by yourself, basically. You're the
Thank you.
perfect guest speaker. And even answered the chat questions and so on. Yeah, if anyone has last comments or any last questions, please ask them. And then, because we've been going now almost an hour and a half, so we have around 5 to 10 minutes maybe left, Howard, until we close the room. What do you think, Howard?
Sure. If anyone has any other questions, I'd be happy to answer them. If not, we can end it there.
May I say something?
Absolutely.
I really, really enjoyed this talk. So it's almost thinking of you can have like a customized library of your, whatever research direction you're thinking of. And then my kind of, I don't know if it's the right way to ask, is that because when you said increase the particular, evaluate a certain percentage of gene expression versus the outcome, because Katrina was asking, can you give some kind of hint at the organism level if I want to alternate like 5 proteins, 5 enzymes, what kind of outcome one might get? And then, right, it's kind of, I thought it's complicated, but I guess in your system may be possible. Sounds like. Thank you.
It's a great question. So I like to call this the curse of dimensionality. So whenever you are engineering any system, whether it's an organism or otherwise, with lots of tunable knobs. So let's say you have 5 proteins that all are doing something you want, but you might need to produce more or less of the protein to avoid, other, bad things from happening. For example, accumulation of some toxic intermediate. So you have 5 tunable knobs, and you don't know where you should turn the knob to. Should you, turn the knob for protein number 3 to 2, 3, 4, 5, 6, and so on, right? So it's like it's an unknown. So we've developed a solution to that problem. So we can design DNA sequences that are what are called degenerate DNA sequences. So for example, if you go to like a IDT or another oligo, synthesis company, you can type in, of course you can type in AGCT, But you can also type in other letters like R or S or W. And when you type in those degenerate letters, you're basically asking the company to send you a tube that contains a mixture of primers or oligonucleotides. And so we use that technology and we actually design degenerate, DNA sequences, so mixtures, that we can then introduce into the organism so that after we introduce those mixtures of DNA sequences into the organism, what we're really doing is systematically varying the gene expression levels. Again, we're tuning those knobs, but we make sure that when we introduce those sequences, we tune the knob from a very low number, let's say 1, to a very high number, and I'm going to say 11 from Spinal Tap, and everything in between. So that with a small number of different DNA sequences, we make sure that when we do the experiments, we've completely tuned those knobs from 1 to 11 and everything in between for all those different proteins of interest. And so when you generate the library of DNA sequences, and when you introduce that library into the organism of interest, you of course get a library of cells, and each one of those cells carries with it a different combination of your system of interest. And each variant will be expressing those proteins at slightly different levels. And then, of course, you have a high throughput assay or a biosensor that you can use then to do the screening and selection. It depends on the product of interest, whether it's metabolite or protein or function. But you are basically searching and sorting through that library to get the best ones. But importantly here, I call it the curse of dimensionality, right? If you were to do this randomly, and we've shown this, if you do it randomly, you basically will miss all the good stuff. That is, when you introduce random DNA sequences, you do not systematically explore the function space. But if you use our models and design the DNA sequence libraries, then you can systematically explore the function space and pull out the best ones from the screening. And we've shown this several times, particularly with metabolic pathway engineering, where we are dealing with large numbers of enzymes that work together. And we show, with some nice experiments, that if you randomly mutagenize your DNA, you don't get great pathways. But if you systematically design your DNA sequences with our models, then you systematically search the expression space, which gives you the full function space. And then actually you can use that data to even understand how the system is working. For enzymes, you can actually use the data to figure out the kinetics, the Kcat and Km, of the enzymes that you're introducing, which is valuable for downstream optimization. So again, with our models and algorithms, you can intelligently design and engineer these systems even as they get really complicated with lots of moving parts. Because again, the more predictions that you make, the more control you have over the outcome.
Right, right. So that's definitely something to look forward to. That the people who use your models, people's research is coming out of that. Okay, thank you so much. No more questions from me.
Very welcome.
Yeah, thank you. I had you talked a little bit about the indicator that you designed to detect explosives. That's really interesting. So which type of indicators are you working on, or are being designed, because in neuroscience a high need would be a potassium, a really good potassium indicator. Like, are people asking you, probably from all kinds of fields, to help design these type of indicators, or is it not yet like a booming, basically, part of your company work?
So we've developed all sorts of different biosensors, including what are called riboswitch sensors, and including a riboswitch sensor for the explosive TNT as well as DNT. And then we utilize that riboswitch sensor for TNT in a Bacillus subtilis organism to detect explosives in the ground. Again, the way that we engineered those riboswitch sensors is by leveraging our model predictions. And so here's an example where we started off developing and validating a model for translation, but then we figured out that we can apply that same model to design and engineer what is called a riboswitch sensor, just by using the model to figure out how to incorporate what is called an aptamer into the messenger RNA. And so our algorithm is called the riboswitch calculator, and it allows you to take any RNA aptamer and figure out how to embed it into a messenger RNA ribosome binding site, and how to design the sequence before and after the aptamer, so that when the aptamer binds to your metabolite or protein of interest, that binding event will cause the RNA to change its shape, and then that shape change will then activate the expression of your output protein of interest. And so we show with many different aptamers. We've done now over 8 different aptamers, and these aptamers bind to small molecules like TNT. We've engineered riboswitch sensors using aptamers that bind to thyroxine as well as dopamine. So we have a dopamine sensor. So if you're interested in neuroscience, and thyroxine is a hormone. We've also developed, in work that we just submitted, we developed a riboswitch sensor for C-reactive protein, which is a biomarker, as well as, let me get this right. Another one, it was interferon, so IL-32 gamma. So that is another biomarker commonly associated with pro-inflammatory response. But again, we have this workflow where we start off with an RNA aptamer that binds to these ligands which I mentioned, and then with our model predictions and design algorithm, we can then generate completely new riboswitch sensors that bind to these ligands of interest, change shape, and activate the expression of these of an output protein. And all of this takes place either inside of a cell or in a cell-free assay. And the benefit here is that it's all genetically encoded. It's like the cell or the gene expression machinery is producing the reagent for detection. And so we see this as a really low-cost sensor. It's the type of sensor that can actually be dried on paper and rehydrated upon sample addition. So it's quite easy to use.
Wow, that's really impressive. Do you think we can build those someday into materials that we wear to maybe detect pollution or, other kind of, with more and more pollution maybe?
Yeah. People have actually taken a biosensor and in a cell-free assay, and they've put it onto a fabric, and then if a sample hits the fabric and hydrates it, it will activate the sensor and report on what's in the sample. So absolutely, but there's, how about this? The best science fiction-y but completely possible application for all this is what I would like to say is a health tricorder, right? So imagine going to CVS and buying a little box, and in that box contain around 100 different tests, and each box costs like $10. So every month for $10, you can on your own in the privacy of your own home you can take a sample, probably blood, and put it into the sample, just like a COVID test, but instead of testing for COVID, you're testing for 100 different metabolites and proteins and other ligands of interest. And so this is possible. And so with this type of assay, it's low cost because the box doesn't contain something expensive like a monoclonal antibody or something like a synthesized aptamer. The box only contains a cell-free assay, which is very cheaply produced, and molecules of DNA that contains the instructions for producing the biosensor, which again, the DNA is cheaply produced. And so just by that assay, because it's just lysate from bacteria with a couple of extra ingredients and a DNA sensor, or a DNA that contains the instructions for the sensor, just by combining those two things, now you've lowered the cost of the assay to dollars per test, as compared to if you go and get a typical ELISA done, it's many more dollars than that. So this type of assay format can be scaled up for quite a few targets, and it can be combined together in a single format so that one sample can then be applied to multiple different biosensors at the same time in a multiplex fashion, and you can actually make this possible.
That is, amazing news. And also, especially when you say in the privacy of your home, because you can decide if you write down the results, if you put it in your phone, or if you share it with some app, because lately there's a
Yeah,
lot of discussion about data privacy, your medical, what happens to all the data you put in your different phones or devices or watches. What happens with it? Do you want that people use it, sell it, whatever? And that way, since it's an analog way of doing it, it's your decision how you store the data, use the data. And I think that's really great, because there were, I don't know if you read about the news that even some hospitals now are getting sued because they sold or used some app, I forgot what it was, on their portal, that then other companies had access to all the patient data.
Facebook
So
Pixel, the Meta Pixel code.
right, it was Facebook had access to all the patient data from a few
oh boy.
hospitals. Yeah, so there's a lot of at least in people that work with data, a lot of fear about losing control over your data. And with the abortion laws changed, where
Yep,
these apps that monitor fertility get some people in trouble. So
absolutely.
yeah, that analog way is the way to go. So thank you so much for doing this amazing work. And it's really always doing these rooms is also very non-altruistic, because it gives me hope for the future to talk with people like you that use their talent and their knowledge for these type of projects. So thank you for giving us hope, and for designing a brighter future. So we wish you all the best and all the funding, and everyone go to the company website and talk about it, so to support Howard, which you probably don't even need, but thank you.
Well, thank you very much. It's you're all very wonderful, great questions. I really enjoyed talking about the work and speaking with you all today.
Yeah, thank you. And whenever you would like to come back, please feel always invited to talk here again. And everyone, enjoy the rest of your day. And thanks everyone for coming. We will if you like discussions like this, follow the room, follow Howard. And we will have tomorrow Dr. Varun Venkataramani talking about how glioblastoma manages to invade the brain. And on Friday, we'll have Dr. Goodyear talking about how he uses antibody dyes to visualize heartbeats in vivo. Yeah, and we'll have more guest speakers the following week. So thank you, Howard, again. This was such an amazing talk, such groundbreaking work. We are honored that you came here today, and we wish you all the best. Thank you.
Thank you very much. Have a great day, everybody.
Clay, we close the room in 3, 2, 1. Bye, everyone. Thank
Bye-bye.
you.